Hi, my name is Jordan Rodriguez, and I'm a postbac researcher at Texas A&M University, Corpus Christi. Over the past 6 months, I've been working to successfully sequence the genomes of fish specimens that have been preserved for over 110 years. One of the greatest challenges facing humanity today is our ability to understand the impacts we have on the natural resources we rely on for our existence. In marine ecosystems, biodiversity not only provides resiliency in the face of climate change, but also protects food security across the world and enables the livelihoods of many people in coastal communities. An improved understanding of the impacts we have on these vital resources could give us meaningful insight on how to better protect and conserve them for present and future generations. One way of understanding these impacts is by turning to the information locked in historical specimens, in which such information can be used to address current societal challenges and help us to answer questions regarding population decline of marine species and their adaptation to years of extensive human impacts. However, sequencing historical samples has challenged researchers for decades due to DNA fragmentation and degradation and enzyme inhibitors present in the preservatives. Despite this, historical specimens have been made available through the Smithsonian National Museum of Natural History for many years, in which an example of these collections originated from the USS Albatross expedition that took place between 1907 and 1909, where over 91,000 specimens were collected from the Center of Marine Biodiversity, known as the Philippines. This expedition to the Philippines resulted in one of the greatest marine natural history collections in history, making these priceless treasures available for present-day use. In our case study, we attempted to sequence the DNA of a Philippines marine fish species that was collected during the USS Albatross expedition, Salarias fasciatus, aka the jeweled blenny. We chose this species for a number of reasons. First, because of its popularity in the aquarium trade, a selective pressure that came to be shortly after World War II, meaning that this species did not face these pressures during the Albatross expedition, but has increasingly done so since the 1940s when the aquarium trade began to take off in the Philippines. And due to there being a chromosome scale reference genome available through NCBI, this species is particularly useful for exploring my hypothesis that the genomes of century-old specimens will be successfully sequenced using low-coverage whole genome sequencing, or ATBC. So, as the cost of sequencing continues to go down, LCWGS has recently emerged as an alternative to other sequencing efforts because it is powerful and cost-effective. The way that LCWGS works is that we can maximize the information we get from the sequence data by spreading it across the entire genomes of many individuals. And this largely avoids the negative effects of DNA degradation and enzyme inhibitors in the preservative. In the figure to the right, we see that in restriction site-associated DNA sequencing, such as RADseq, small portions of the genomes of many individuals can be sequenced deeply. And this has been one of the most popular approaches for studying population genomics. However, one limitation of there being large stretches of the genome between markers that are unsampled means that this type of data may miss signatures of selection, which can be highly localized in the genome. Because of this, genome-wide coverage is often needed to understand different aspects of population demography. In the middle, we see that sequence pools of individuals is a good way of obtaining estimates of population level parameters, but all information about individuals is lost, making it difficult to control contribution to the pool or to perform downstream individual level analysis. This being said, we sacrifice depth of coverage for breadth of coverage and thus work with genotype likelihoods under a probabilistic statistical framework rather than called genotypes. To do this, we obtained 97 specimens from the same location, 36 in which were historically collected aboard the USS Albatross expedition, and 64 in which were contemporarily collected by the Philippines Pirate team in 2019. We then extracted the DNA using the DNeZ tissue kit and constructed libraries for each individual. We then sequenced Illumina paired-in short reads in which we performed sequence quality trimming, adapter removal, filtering, deduplication, and decontamination of the raw data following the Garcia et al. Pipeline. We then mapped those sequences to the reference genome using BWA-MIM2, in which we then used those mapped sequences for our downstream genetic analysis. We utilized PC-Yanks to estimate the covariance matrix and individual allele frequencies for low depth NGS data, in which we outputted different principal component analysis that I will share with you shortly. In the right, we see that a pattern in which we have two distinct groupings would be a signal that these are individuals from different populations or different species. In a pattern on a PCA where we see overlapping points, we see that that is likely because the individuals are from the same population and same species. We then ran ANKS and NGS relate to infer relatedness of RXY, inbreeding coefficients, and other summary statistics to further confirm the successful sequencing of the century-old specimens. In the box to the right, we see that identical twins would show an RXY value of 1, while distantly related second cousins would show an RXY value of 0.0325. This is important to keep in mind for some of the downstream results. So with one of the greatest challenges for scientists being deciding how to distribute sequencing efforts, we wanted to see the sequencing efficiency for our historical samples. We see here in this figure that fewer historical sequences mapped for the historical samples, and thus meaning that more sequencing effort was needed for these samples to reach our desired depth of coverage. In the figure to the right, we see that we overly achieved our goal in that we have equal or greater than genome coverage and depth in the historical specimens versus the contemporary specimens. When we ran PC-Angst, we outputted PCAs, or principal component analysis, for each chromosome for both era types. In this figure, we see that there is a strong similarity in 5 of the 22 chromosomes of structural variation, which we believe is a sign of chromosomal inversion. The important takeaway here is that this structural variation has persisted through both era types. Furthermore, in the rest of the chromosomes, we saw a pattern similar to this one, where we don't see any sign of chromosomal inversion, but we still see this similar pattern between the points, signaling to us that we have indeed sequenced the DNA of the same species from the same population. Now, to confirm that we sequenced the DNA of the contemporary samples, the historical samples and not contamination from the contemporary samples, we ran relatedness analysis. We see here that between any two individuals, the relatedness coefficient of RXY does not exceed the value of 0.025, in which, if you recall, that means that none of these individuals were closely related. And if the historical sequences were a result from contemporary contamination, we would have expected to see a very large RXY value. So this finding confirmed to us that we did successfully sequence the DNA of our historical samples. So for the discussion, we successfully sequenced the whole genomes of 32 out of 36, or 89% of the samples from the Smithsonian's extensive 110-year-old albatross collection at a mean depth of about 2x coverage. We also find that LCWGS is less expensive than mtDNA barcoding of historical specimens, coming in around $30 per fish. And for our next steps, We plan to carry out additional historical demographic modeling, including looking at things like linkage disequilibrium and changes in the effective population size of this species to test for population decline in response to aquarium trade. So with that being said, these are my acknowledgements, and I would like to specifically thank the Philippines Power Project for funding me here in the Philippines to conduct this research, and also to the NSF for providing me the grant in which I work under, as well as Dr. Christopher Bird, my PI, and everybody else listed on this slide. Here are my references. And thank you all for tuning into my talk, and I will now be taking live questions in the chat.